Before I get started, for those of you that have seen me give a talk before, I want to introduce everyone to Biscuit. Biscuit is a golden retriever puppy animal that my daughter, who is now 6 years old, has had since she was born. And whenever I travel abroad, I take Biscuit with me so that she can see where I've been. So before we talk about science, I want to take a photo of everyone saying hello to Biscuit. So if you can wave, then I can show my daughter where I've been. Thank you. Perfect. All right. So it's always my favorite part of a talk to introduce Biscuit. All right. So my title has changed a little bit. I'm going to be focusing specifically on the genomics of genes in the immune system today, not whole genome sequencing as you just heard from Tomi, but I think it's critically important and it complements the message that he just gave us by that stupendous talk. I'm not sure which direction to look, so I'm going to go this way. All right, so the work that drives us in our laboratory and has driven me for my entire career is trying to understand why we all respond differently in the context of infection or vaccination, why some people develop autoimmune diseases and others don't. So a great example of that is the contrast between the seasonal influenza vaccine, which in the US can sometimes be only about 30% effective, versus a lot of the recent mRNA vaccines that were developed for SARS-CoV-2, which are up to 90% effective. Understanding why their differential response usually ends up being a question of the formulation of the vaccine itself. Did we choose the right virus for the vaccine? Do we have a proper adjuvant? But very little has actually been considered about how the host and the genetics of the host may influence the response to these vaccines. And this is critically important because many pharmaceutical companies are now focused on developing what they're calling universal vaccines. If we all are different and that affects our response, then it doesn't matter what the formulation of the vaccine will be. And so we want to make sure, again, to the point that Jeff made and others, that each person is being treated with something that takes advantage of the diversity in their genome to have the best response in the context of an infectious disease. So a great example I give of this is the measles, mumps, and rubella vaccine. Over 3 million children get this vaccine in the US each year, but up to 10% or 300,000 children will not respond. But only 20% of that response is understood. And work that has come out from our labs in the last few years has demonstrated that the genomic background of these individuals may influence that immune response potency, and that's what I want to talk about today. So I'm going to talk about antibodies. Many of you probably don't think about antibodies on a regular basis, so I want to make sure that we are all starting from the same foundation. So antibodies have a huge role in the immune system. It's half of the immune response, the humoral response. This is what a schematic of an antibody looks like, right? It's a canonical Y shape. The business end of the antibody is up at the tip of that Y. That's where the antibody binds the antigen, the virus, or the immunogen, and can neutralize viruses or coat them and opsonize them. However, the stem of that antibody is incredibly important for function. And as I hope I will convince you today, it was called the constant region because it's far less variable than the variable region at the tip. But that does not mean it's actually constant. And variation in that constant region can actually influence response to vaccination and to autoimmune development. So for those of you that took immunology in university, I don't want to trigger any stress responses. VDJ recombination is very complicated. We're all taught that we have these genes, that they're all the same across everyone in this room, but we just mix and match them differently to generate our antibodies. This is an oversimplification of this locus. This locus actually looks like this. So rather than a few V's, D's, and J's, there are hundreds. They're confounded by the integration of pseudogenes, and it is the combinatorial diversity of this that generates the 10 to the 12th different antibody specificities you can have in your immune repertoire. Now, to the point of the talk that I was invited to give of why we need long reads for this locus. So these genes are approximately between 200 and 400 base pairs in length. They're highly repetitive. 50% of them lie in a large structural duplication, and so short reads cannot map this locus, and I'll show you what that looks like. But if we back up even before short reads and think about microarrays and GWAS studies that for the longest time dictated which genes we studied in the context of disease. So what I'm showing you here is the Illumina immunoarray coverage of SNPs across the human genome. So we all know that HLA is associated with almost every disease. A lot of that was due to the happenstance of the fact that oligonucleotides covering SNPs in the HLA was included in these arrays and then used in GWAS studies. 9,000 SNPs to be precise. Even in the locus that control or encodes the NK cell receptors, the KIR locus had over almost 1,000 SNPs. In the immunoglobulin locus, there were 5. So all of those GWAS studies for a decade that we used to draw associations of disease with polymorphism at the population level would not have seen SNPs in IGH because it wasn't included in the array. Why wasn't it included in the array? Because it was part of the genome that was not complete until the telomere to telomere genome in 2022, which required HiFi reads. This was part of the original Human Genome Project that dropped out this end of chromosome 14. There was no coverage here. In fact, the first assembly of this locus wasn't published until 1997 by Dr. Matsuda at Kyoto University, and there was only a second reference published by my colleague Dr. Watson in 2013, and up until the T2T genome in 2022, there were only 2 references for the entire world. And so we had no idea what the population diversity of this locus was like. If you try to sequence it, target it, and sequence it with short reads, it's incredibly problematic. So this was done by Dr. Watson during his reference generation of the second reference. He sheared genomic DNA to small fragments, 400 base pairs, enriched them with oligonucleotides specific for that region of the genome, and tried to assemble them. So using paired end reads off of a MySeq, you end up with this stack of piled up reads that you can't tell. Is that a bias from the capture? Is that a duplication? Is that just standard coverage? The coverage is so patchy that you can't see if there's structural variation there. Now, I don't want to make the PacBio folks in the audience cringe because I'm going to show RS2 data. So everyone just take a deep breath. This is data that's actually generated in 2016. But even then, with 6 kb reads that were highly error-prone on the RS2, which is not the way it is today, right? Revio is incredibly accurate. But we could resolve a copy number variant here that was never able to be seen with short reads. So even back then, the data was only capable of being done with long reads. Since then, so this is Corey Watson and I, so now Corey and I have developed what we're calling the SWATSON labs, so the Smith and Watson labs, are really focused on trying to understand how genotypes in these immune receptor genes impact the immune receptors that you express and therefore the protection you have against disease. So we've developed 2 new molecular tools. I really only have time to talk about the genotyping tool today. I will mention the RNA tool in passing. If you're interested, please come find me. I love talking about this stuff. So what we do is we use the same approach I just talked about, but instead of capturing 400 base pair reads, we capture 10 to 15 kb reads. The only restriction on this length is that because this is an enrichment protocol, there is amplification, so we can't go bigger. But that is really the nice sweet spot for PacBio sequencing. When we do that, we can multiplex multiple individuals per run, which brings down the cost. We have incredibly high accuracy, so these libraries have an average of a Q42 or over 99.99% off of the Revio. And in fact, this initial assay was developed for the Sequel IIe. We ran 12 individuals per Sequel IIe. Now we can run 36 to 48 on the Revio. So we actually have increased our throughput in the last 2 years, or the last year, I guess, from about 3,000 samples a year to about 10,000, which is fantastic. So what does that data look like? So this is an overview image from a paper that we published last year in Nature Communications. This was the largest cohort at the time, 154 individuals from multiple worldwide populations. Do I have a pointer on this? No, whoops. All right, so what I want to point out to you is on the top section, you can see all the different V, D, and J genes that are included in this locus. Below that, you see the column that on the left side is labeled SVs, or structural variants. These are structural variants that were found in at least 5 individuals of this 150-person cohort. And what you see is that there's a variety of structural variant patterns that include massive deletions. So the red dots represent pieces that are removed from these individuals' genomes. And when they're removed from the genomes, they actually remove, obviously, those genes. So that makes them no longer available to be used in immune responses. We also see insertions represented by those inverted triangles. So these are the addition of genes that some populations have and others do not. Additionally, in addition to just structural variation in the presence or absence of genes, these genes also come in several different flavors or alleles. Some genes are enriched in widely diverse alleles, and you can see those on the bottom. So in 154 individuals, we found 127 new coding alleles. So these are productive alleles to be used in immune response that may differ by population. And we did find a very population-biased distribution of this genetic variation. People within an ancestral population are more similar to each other than other ancestral populations. If you look at the pattern of deletions in the individuals in our cohort, I just want to draw your attention to this outlier individual. These were healthy individuals, so to some extent, Dr. Pattinson, I think there is some use in sequencing healthy individuals, especially in these areas that have not been surveyed in the past, just to understand how much diversity we're dealing with. This individual, apparently healthy, donated their blood to a commercial biobank, but walking around with 30% of his locus deleted, 25% of those genes missing. So if you were to design a vaccine to target the genes that this person is missing, that person would not be able to respond. And that's critically important. So why does genotype matter? So genotype matters because it dictates what we can express and what is encoded into proteins and has function in our body, right? So our next step was to determine the impact of these variants on the antibody repertoires. So if you think about this blue gene pattern on the left, obviously if you have a gene deleted, it cannot be expressed in the repertoire. But one of the advantages of our method is that we sequence the genomic DNA. So we get variation in coding and non-coding genes. And when we do that, we can also see variation in the non-coding regions that impact the regulation of expression in the antibody repertoire. So in the case of those orange genes, if you have that A SNP, it may be in the middle of a regulatory sequence that keeps that gene from being expressed, even if you carry it. So overall, in those 154 individuals, we found thousands of SNPs, hundreds of insertions and deletions, and tens of very large structural variants. And in this paper, you can read in detail how they impact our repertoire. We found, in fact, that 70% of the genes that encode our antibodies are dictated by genomic variants or regulated by genomic variants. This is completely different than that paradigm that I shared with you in the beginning, where we all have the same genes and we mix and match them differently. That's not actually true. What it means is that the foundation of your genomic structure dictates what you express, and that we should be understanding what that is when we're designing sort of next generation drugs that are most effective on a population basis. So let me tell you what I mean by that. This is a great example. As it was mentioned, my degree is in virology. I do a lot of work in the HIV space. The current forerunner of HIV vaccines is actually this synthetic trimer that is targeted at this gene, this very specific variable gene, V1-2. If you have the star O2 allele of V1-2, 20% of your B cells express this receptor. It is ready. It is ready for that vaccine. It is ready to respond. However, if you have the star O5 allele of the same gene, and I will show you the star O5 allele differs by a single residue, then you have less than 1% of your clones that carry that. And it's hard for me to imagine a world in which a vaccine that is given in your arm would find the one B cell in your toe that has the right receptor, right? So the frequency itself may impact your ability to respond to that vaccine. As I mentioned, these are the star O2 here is the reference, and the other alleles are listed below. These alleles differ by single residues. These aren't dramatic genetic variation, but those single residues make a huge difference in how those genes are expressed in your repertoire. So this is RNA sequencing data. If you have the star O2 allele, it's very dominant. You have high expression of those receptors in your antibody repertoire. But if you have that star O4 allele, you have 5-fold less, which likely will impact your ability to respond. In fact, we've taken this further in a new cohort in collaboration with Bill Schief, who's at Moderna now. We did an additional experiment with this vaccine, looking across a wide variety of genotypes. And if you have the star O4, star O5 allele, you actually have much less expression, as I've already shown you. So there's actually a dose response based on the genotype you have if the proper antibodies are expressed in your reservoir, or in your repertoire, excuse me. So And the star 05 allele is actually worse than the star 04 allele. If you have the star 05 allele, it's a very hard to envision how you're going to respond to that vaccine. And not only is it a frequency issue, right, because we can all imagine that that B cell will somehow get to that vaccinated B cell or vaccinated lymph node, but if you actually engineer these mutations into the good allele and add back that arginine that is the difference between star 05 and star 02, and then do a protein-protein interaction assay on the BIACOR instrument, right? Surface plasmon resonance, looking at affinity for these interactions, you actually no longer bind the antigen. So not only is your B cells incredibly rare, but if they do find the immunogen, they don't respond. They've lost their affinity, they cannot bind the immunogen, which means they will not blast, they will not respond, you will not get protection. And the authors of this study state that this is not precluding that there could be a response, right? This antigen may hit a different antibody gene, but it's not the way it's designed. And that may impact it on both a frequency level and an affinity level. And that's dramatic for the potential of how the people who will not get protection from this vaccine. And what I'm really excited to show you today, I've never shown this data in public actually, is that we took this information and we went back to our population cohort, and in 380 individuals, oh no, this is my old deck. I had that one slide that I added that was really exciting. Hopefully we can come back to it, Jiwon, or should I move forward? Okay. What I wanted to show you is how this allele is distributed across different populations. Maybe we can load that up before the end, because I changed my slides just before the presentation specifically to show that, because I'm so excited. Anyway, so what we've done is to figure out where we think would be best to start applying this in disease, as opposed to just sort of healthy donor cohorts and what we think of as sort of collecting genotypes. And where we started was myasthenia gravis. So this is an autoimmune disorder. It targets the acetylcholine receptor in your neuromuscular junction, and it affects the ability for you to breathe, for you to swallow. You mostly see it initially with a drooping of the eye, and it can be very severe. And the reason that we chose this as a place to start for the clinical importance of the work that we're doing is because it's known to be mediated by IgG1 and IgG3 specific antibodies. So something that requires high resolution at a nucleotide level of regions of the antibody that you can't see with anything except long-read sequencing. What's really important is that right now this disease is more of a syndromic collection of diseases, but there's actually 3 distinct pathological mechanisms. Autoantibodies target acetylcholine, either force receptor internalization, block the ligand receptor interaction, or fix complement. And complement is Fc mediated. If you remember that Y that I showed you of the antibody, the stem of the Y mediates the complement fixation. And this is something that isn't routinely screened in antibody profiling. Usually antibody profiling with short reads only screen the variable region or the arms of the Y. We have a method, again, I didn't have a chance to tell you about today, but FLAIR-seq, which does the full-length heavy chain and sequences the polymorphism in the stem as well. So in this cohort of acetylcholine myasthenia gravis individuals, we have 48 samples from the Rare Disease Network and funding from them, thankfully. So we did the DNA genotyping, as I've described to you today. We also did the FLAIR-seq repertoire profiling, which we'll talk about just slightly. And this is what we saw. So first off, we looked at those variable genes, what's been studied before, and saw that our method identified signatures in V169 and in V434, which had been seen previously. So our methods recapitulate existing data. And with our RNA-based method, we can tell you in which compartments those genes are expressed. Are they expressed in IgG1, 2, 3, and 4? One of the really big advantages of having that constant region is that we can then look at just the pathogenic subset of antibodies. So we can say these are IgG3-mediated disease. We can remove all the other noise from that analysis and just focus on the IgG3-mediated gene carriage. And then we also looked at genotyping. So what was striking to us in 48 individuals is how much there isn't known. So we're looking here at the constant region genes. They're encoded by these individuals, only 45 people. The blue bars represent alleles that are known in the database. The red bars represent alleles that have never been seen before. So over 80% of the alleles we're identifying have never been seen before in populations, and this is critically important, right? Because if variation in these alleles are what's leading to disease, then current methods are not going to be able to predict that. We've been able to confirm that in our population-based cohort. So as I mentioned earlier, we're very interested in how this genetic variation is distributed across the world. We got a grant in 2023 from the NIH to do 3,500 samples from 58 world populations based on a Sequel IIe pipeline. Now, with the acquisition of the Revio, we can sequence over 10,000 individuals for the same price because of the increased throughput. The data is more accurate, the turnaround is faster, and so we're really excited to be here and trying to build collaborations to really expand our population survey around the world. So back to the story at hand, in the context of myasthenia gravis, the most novel alleles were actually found in IgG3 and IgHA2, which are known to be involved in the disease. And so our current efforts are really to annotate these constant regions. A lot of work has been done in this region using antibody engineering to try to identify variants that are more functional in the context of engineering antibodies for treatment. And so we've been able to use that literature to identify specific residues that may be impacting certain individuals' propensity to develop autoimmunity. So there's this great paper by Goldberg and Ackerman that really details the variants in the constant region known to be associated with complement activity. We've compiled additional residues known to be involved in FC receptor binding and post-translational modifications that may impact function. And so what we wanted to look at was whether we saw these variants in our myasthenia gravis cohort. So the first one we really gravitated to was this hinge deletion. So this is a multi-sequence alignment of the known IgG3 alleles. The 5 at the bottom with the star are alleles that were new that we discovered. And if you look at the distribution of this deletion of the hinge, you can see that there are certain alleles that contain that deletion and many that don't. And it's been shown in the literature that this hinge allele deletion is associated with higher activation of complement and more severe disease in the context of SARS-CoV-2. So we looked in our population. Not only did we find the major allele, the one with 4 hinges, we found the short allele, the one with 3 that I just showed you, but we also found this E allele. Hilariously, I think it's called an E allele because my student Easton discovered it, so he got to name it after himself. And this only has 1 hinge. And when we looked at the distribution of these structural variants in the repertoires of these individuals, we found something really striking, and that is that the MG individuals are enriched in these structural variants. In fact, we found a completely wonky variant. Not only does it contain a hinge deletion, but it's got a duplication both in IgG3 and IgG4. So this person has multiple alleles to work with when they're responding to an infection. Now, it may not be striking to you in the audience, but this is incredibly striking to us. We didn't filter these MG cases based on their pathogenic mechanism, right? And 2 out of the 3 pathogenic mechanisms are non-FC mediated. So now we're going back through the data, identifying which bin each of these patients lies in, and we want to look at the distribution of this deletion in those patients for which complement is the main pathogenic mechanism. And I think that what we're hoping, right, is that there's these 3 mechanisms. Those are actually 3 distinct diseases. And the first line of therapy for these patients is complement inhibitors. For those 2/3 of patients that don't have complement-mediated disease, that's an irrelevant treatment. They shouldn't be getting that. They should be going on to the next line of treatment. Then maybe we can use this genetic test to identify genetic variables that are influencing which mechanism of disease they have, identify the patients that would be most susceptible to responding well to complement inhibition before going on to more severe treatments like rituximab, and then further use that genetic test to define, do you have complement myasthenia gravis or blocking myasthenia gravis, and try to really delineate which disease people have to line them up to their best treatment option. So I think the other thing we want to show, of course, is that this deletion just isn't in the DNA. It's actually expressed in the repertoire, and here you can actually see just a line reads showing that there are repertoire molecules that contain that hinge, so it is moving through the canonical molecular biology pathway. The last thing I want to mention is that this is not just an antibody thing. So for those of you that do immunology, you know that B-cell immunologists never really talk to T-cell immunologists. And what I think is really important is that this is also a T-cell thing. So here is genotyping that was done on 9 individuals from the 1,000 genomes cohort on the T-cell receptor loci alpha, beta, and delta. In 9 individuals, we found 79 new alleles. So this idea of incredible variation in these immune genes has not yet been recognized, that is only resolvable by long reads. We're just scratching the surface. And so any of these boxes that you see that are red with a dot, those are novel. Anything that's blue with a triangle is a deletion. So you also have that structural variation here that, again, only can be seen with long reads. This is critically important because, of course, T-cell responses are essential for proper response to immunotherapy in the context of cancer and other treatments. And so understanding how genotype may impact immunotherapy responses is something that's a very active area of investigation for us in the lab. So with that, I'd like to wrap up. So the most important thing I want you guys all to take home today is that immunogenomic variation matters. And more than that, data diversity and representation matter. I think that's why I'm so honored to be here today and was so excited to accept the invitation from PacBio. I've never been to Thailand, but all of you out here from the APAC region in the audience, you are the ones that are expanding the representation of different populations around the world. You are the one who are taking genomics to the next level and allowing us to understand how we can leverage the diversity that's in all of us to make better therapies for everyone. And that's not going to come from a single reference from one old Caucasian guy in the US, I promise. So I think it's really important that we all are building collaborative networks, that we're all working together and showing that we can leverage the genomic diversity to make human health better rather than ignore it. I think we've just started to scratch the surface of immunogenomic variation, how it impacts health, disease, and response. This isn't just an antibody issue, it's definitely a T-cell issue as well, which means it has very wide-ranging applications in both infectious disease and oncology. So in terms of next steps, I'm going to take one little moment here to do a plug for myself. Because we're so excited about the potential of this, because we think that it has incredible application across a bunch of areas. Corey Watson and I, together with the additional team, have founded a new company. So this is the first public announcement, I guess. I'm very excited. We've been founded for about 2 months, but we are Clario Biosciences. So if you are working on something where you think that understanding this immunogenomic variation would help in the context of your disease cohort, come talk to me. We offer this as a service. We're looking for partners. We're very excited. So I'm going to use my PacBio time to plug my Clario company. But I'm really honored to be here today to talk to all of you. I would love to answer questions and discuss more. I can't not thank my team. This is us all being very excited when our Revio was delivered. I'm very lucky to have built such an incredible foundation for this work in Louisville, Kentucky. And with that, I'd like to thank you all for your attention. Maybe while we're answering questions, we can slip in that slide. Oh, yes, wonderful. This is the slide I was so excited to show you. So I've never shown this in public. So if you think about back to that HIV story I told you, where you have that SARS-CoV-2 allele that's so important for response, this is the distribution of those alleles across populations. So this is a very rough preliminary analysis. There's only 380 individuals here. But the pink, that's the SARS-CoV-2 allele. So you can see in the European population, which is the 4th group over, that pink is very dominant. Anything that is yellow and green and blue are the alleles that are not responsive to that vaccine, right? And if you look in the East Asian populations, which is in the 3rd section, there's a significant percentage of the populations who carry, quote unquote, the non-responsive allele. If this is the vaccine that is the furthest ahead in clinical trials, it has the dramatic potential to not work for the people who need it, right? This HIV is now a disease that disproportionately affects the global South and Southeast Asia. It's incredibly important that the vaccines that they're being produced in the West actually help the people that need it. And so this type of understanding of population diversity at these loci not only help us hopefully predict who will respond, but also inform vaccination makers, immunotherapy developers about what types of strategies are going to have the most reactivity in the largest breadth of populations. And I think that's critically important. So with that, thank you very much. Thank you for loading my slide. I appreciate it. I'm happy to take any questions.
Thank you. Could you please remain for anyone want to ask the questions at this moment? Please.
A very nice talk, Dr. Smith. Also, this is IPM from Berry Genetics in China. I have two technical questions. It's very amazing to me that about 10 to 15 kilobases can be obtained after hybridization capture. So my first question is that how frequent is the probe designed against to the reference, to the target genome?
Sure. A lot of that is not my part of the collaboration, so I don't know all the details, but we do have probes that are designed against the entirety of the immune receptor loci. So in this case now we're running 7 loci. So we run the IgH heavy chain, kappa, lambda for antibodies, and then the TCR alpha, beta, delta, gamma for the T cells. And so the oligos are the standard 120 base pair baits. We did spend 10 years optimizing the protocol, so I agree with you. It was not the easiest thing to do. It's taken a lot of work, and there's a lot of finickiness in the protocol, but it's working quite robustly now, so we're excited.
Okay. So my second question is, how is the size selected?
Right, so we do use the Sage Science size selection equipment. So we size select after the genomic DNA is sheared. So we shear to about 15 kb, and then we capture any fragments that are larger than 8 kb for our capture and sequencing downstream.
Okay, thank you so much.
Absolutely, my pleasure.
All right.
Oh, there's
Okay, please,
here, yeah.
please go on.
Yeah.
เชิญเลยค่ะ
Thank you very much. Very nice presentation. Herawati Sudoyo from Indonesia. I'm just wondering, actually, Indonesia is very diverse population, and you know exactly your statement regarding whether the vaccine will be useful for our population, and yet we have experience with some vaccine which is actually related to the region of our country. So you mentioned about the immunoglobulin, yeah? And we actually try to look at the VDJ, but I think your finding actually supporting or give an added value to our population. So just supporting or would like to, maybe we can talk about it later. And what about the T-cell response? I think that's also important. What about it? What about the Asian?
Absolutely. So I feel like if I had planted a person to ask me a question, you asked me the perfect question. So thank you. So to your point first, the first part of the question about VDJ and understanding immune responses, what we have learned in the process of this project so far, and I think we're just starting, is that because every time we sequence anyone from any background, Caucasian or whatever, we discover something new compared to the existing databases. And what that shows us is that the databases are truly awful. I will use that word. So that means that if you were using standard protocols that use short reads to profile variable regions, which are used around the world, and there's nothing wrong with those protocols, but you have to map them to a reference, right? You have to identify what genes they are. Sometimes you impute the germline from these data. And then the most important signatures, right, are these rates of mutation and things like that. Because the databases are horrible, we need better references. Because right now you can say, oh, I discovered this gene, it has 20% mutation, and that's why it's responding or not responding. When in fact, if you were to generate the genotype with our technology, that 20% mutation is wrong because they have a novel allele you haven't seen before that accounts for 10% of that variation. So because so little has been done, because the technology wasn't available until recently, to generate these precision, I hate to say personalized because I don't think it's scalable to do this for every person at the moment, but especially population-specific references, pangenomes per population for this locus, to represent the actual genetic difference that is there. It makes the ability to interpret the repertoire profile incredibly difficult. And so one of the things that we're striving with our academic projects is really to generate those 10,000 samples that I told you about. Those are to generate references for as many global subpopulations as we can find collaborators to provide and work with. It's really important to get as much representation as possible so you can have the most accurate interpretation of your repertoire data. For the T-cell question, I completely agree with you. And we are only now starting to get into this space with the generation of those 9 references. The idea, again, that we had 9 references with 80 new alleles really shows us how underexplored this is. There have been some studies that suggest that genotype impacts the development of immune-related adverse events in immunotherapy. So we are actively looking at whether you can use TCR genotypes to predict response to certain immunotherapies, to help advance clinical care and choose the right path for different individuals. This isn't, again, just about oncology, although that's a really active research space, but also about T-cell responses and vaccination. So all of those things are ongoing. I would love to talk with anyone further about potentially collaborating. This is such a rich room for me to learn from as well. So thank you all for your attention. Very excited to discuss more. Thank you.
Thank you for your questions, and also thank you, Dr. Smith.